Yes, here I am again. Hi everyone. Hello and welcome again. I'm Oliver, Microbehunter here. Welcome to another microscopy live stream. Just like every week, just give me a second or so until I get everything sorted out. I think it seems. Yes, it seems to be working. Usually there's always a usually half a minute delay when I say something until you are able to see it. And for this reason, I always have to wait a little bit. But I think we're ready to go. Okay. Hi everyone. Basically today, just some water again under the microscope. I did find a few interesting, hopefully interesting microbes. However, not only that, I would like to also mount them, make a permanent mount of those water microorganisms using a mounting medium, which some of you probably already know. It's called glycerine jelly. It's been a request that basically I also make some permanent mounted slides using water of water organisms, and that's what I'll try to do today. Okay. Usually, the first couple of minutes, I go through the chat. Many of you already started to post. I would like to say hello all around the world. It usually takes a couple of minutes for people to join in. That's why I would like to always give it a couple of minutes at the beginning. Yeah. So hello from Vietnam, from Texas, the United States, from Rhode Island, the United States, Connecticut, the US, England, Michigan, United States, Athens, and Greece. Good video, good audio. That's very good. Thank you. Vietnam again, London, Georgia, United States, UK. Okay, Netherlands, Romania, Netherlands, okay, Hanoi, yeah, and Poland. Hello from Germany. Okay, very nice. So for those of you who are maybe new to this format, please feel free to comment. Okay, I'm going to change this from top chat to live chat. Okay, so that essentially I have the correct sequence of chats here. If you have any specific question or comment towards me, please indicate this with @Oliver or @Microbehunter, so it's easy for me to actually see the chats that are directed to me. Usually after 1 hour, what I will do is I will try to answer some off-topic questions. But feel free to post anything. And if you have any suggestions as we go along, I'll try to pick up on that. Okay. So what I'm going to do right now is I'm going to show you a short video that I made today in the morning. At my time, because I would like to show you how I collected those samples here. Because every week I have to think of a new topic, and in the previous during the previous weeks, I did some samples preparation. Last week, I collected some face mites and so on. And today, I decided to do again something more of the traditional, of water sampling, so to say. And I would like to quickly show you a video and comment on that, what I've actually done. Nothing totally unusual here. Okay. So that's basically the fountain that I visited. It's always my go-to place, so to say. And people come here to actually collect some water because the water quality is quite high. Where there is water, there is life. Of course, and luckily in here I found this sludge. Yeah, those bubbles that you see, these are oxygen bubbles made by the algae by photosynthesis. You can actually see that there's some bubbles there, right? So that's basically oxygen. And over here, usually this was full with algae, always. However, they cleaned out the fountain, unfortunately. So it looks fairly clean, actually. But over there in this little crack, I found this sludge, right? Again, here you see the bubbles quite nicely. So these are basically, it kind of shows that lots of photosynthesis is happening. I wonder who that is. I simply used some tweezers and collected some algae there. There was a lot of moss growing around as well. I did not want to collect so much moss, just wanted to collect more of those algae. And basically that's pretty self-explanatory, right? And that's maybe the new thing. Look what I've done here. Right? I mixed everything because I wanted to rinse off any microorganisms and single-celled algae that can be found in there. So I just put it back again into the water, and look how it looks like. Very green, and then I filtered it. I'm going to show you this filter as well, and simply to allow the smaller organisms and algae and so on to go through, and I simply wanted to separate it from the larger ones. This is essentially not a very fine sieve. It's really important. So now it kind of repeats again the video. Yeah, so it's not a very fine sieve. It's I can show it to you just in a second or so. Yeah, this is actually I was a little bit worried today in the morning when I visited this place because I was already concerned that I was not able to find any samples for the live stream because usually it's full. It was really it's really overgrown with algae, but as I mentioned, they cleaned it, right? Okay, so that's basically yeah how it looks like. There was some moss here on the side as well. Yeah, I was not so interested in that, but that's the the good stuff, right? So yeah, just wanted to show this to you. So this was basically just a couple of hours old. Yeah, so yeah, it repeats again. So we go back to the desk. We just want to show you the sieve a little bit. Yeah, so that's how it looks like. This is actually from yeah an aquarium. I think what they're yeah doing this was part of a kit, I think, for children or so. If you want to grow your own brine shrimp, if you've heard of those so-called the sea monkeys, yeah, you can buy those eggs and then you can hatch them. There's a sieve in here. However, it is not quite as fine as for a plankton net. So it's fairly coarse. That's important. So yeah, all of the microorganisms and so on could actually go through here. So I was kind of repurposing this. If you want to make this yourself, you can also do that, and you can essentially use some kind of a nylon stockings. The ladies' stockings, yeah, those nylon stockings, they actually also work. And I've actually also used those ones. So you can basically try to make something like this. I don't know. I would probably suggest that you take some kind of I don't know, some kind of a plastic cup, a yogurt cup, or something like that, and then kind of stretch some nylon stocking around it, and you can make your own sieve if you want to, right? So, however, there's one thing that interesting that happened. So that's basically the original sample. Yeah, and look what happened. That's the filtered one, and I'm not going to agitate this. I'm not going to move it a lot, just very carefully. It looks fairly clear now, so it's been standing here now for a couple of hours. And if you just go back, I'm going to show you the video again. I'm going to show you the video over here. Let's go forward here. It's forward. Do you see how green it actually looks? Let me show you. I don't know if you can see. Yeah, here, let's pause it. Do you look at the glass jar on the bottom, right? It looks slightly greenish. It's now completely clear because all of the microorganisms and everything settled to the ground, to the bottom of the jar, and that's basically where we are right now. And I, of course, gave it a trial, and I'm going to show you right now, and it's full of microorganisms. Yeah, so I was quite happy about that because normally it's kind of you have to centrifuge it. Actually, I also have done that. I've actually centrifuged this as well. Yeah, so I've taken some of this water and then I was spinning it down. It's not even necessary because it's fairly concentrated here. So what I'm going to do right now, so that you understand a little bit of what the task today is I'm going to of course observe them live, but I'm also going to put some of the specimen here on a microscope slide, and I'm going to dry it so that I can make a permanent slide. And drying takes a couple of minutes time. So for this reason, I'm going to do this first. So because there is a waiting time. Okay. So very carefully, I'm going to move this to the side, and you know this already. Look at this thing here, right? This is my coffee warmer. And I'm going to be using this usually about 55 or so, to place a slide on here with a water sample simply to dry it so that I can later on make some permanent slides. So I'm just going to put, I don't know how many I'm going to put on here, maybe 3 or 4. And yeah, look, what I'm going to do now, look, I'm going to use this here. I'm going to show you. Look, I've made this here. Let's carefully move this away, not to agitate it too much. So that I simply know where the center is, and I'm just going to add a drop of this stuff. And what you want to do, or what I've done, is I've taken over here a sample from the very bottom, especially there where it's green. Okay, so this is where and you carefully take up a small sample here. Okay, and Yeah, I think that should be enough, right? And then I'm going to place it on here and I'm going to dry it. So maybe it's going to be a little bit too hot, so I'm going to go down to 40 degrees. I don't want to yeah, make it too hot. Yeah. As a matter of fact, I think it's actually 40 degrees. I think it is too hot because the temperature is not really kept the way it should be. So let's put this on here. Okay, maybe that's going to spread out the temperature a little bit. So let's take another sample here. Later on, we're probably going to make some more. Okay. And here, and then the point is the following. Honestly, I have mixed feelings about making permanent slides of water organisms, and the reason is because I think they look much better alive and not on a permanent slide, because the movement and everything adds so much more, right? But I'm going to, so that just that you understand a little bit how this looks like, and I'm going to carefully now move this to the side again, yeah. And later on we're able to, yeah, after it's dried, now I can actually go up a little bit, maybe to 55, because of and now let's try to make a sample here. However, I want to show you something. Where is the other one? I want to show you something here before I do that. Over here I have got a new slide box, and I'm going to show you now. Yeah, so that's a new slide box with some new slides. Okay. And what I'm going to do now is I'm going to put one of those here under the microscope as is. I just want to show this to you because this is kind of frustrating. Okay, so let's turn this up. Yeah, it takes a little bit, some time for... Here we go. And look at this. Look at all of those. So there is a lot of, these are all, how do you say, deposits that are on the slide. And this is a new slide. Okay. Look at this. This is horrible, right? So in order, so what I had to do, even though the slide was actually new, I had to clean it. And what I have done is I've cleaned it with some acids and vinegar. The reason is because these are most likely calcium carbonate deposits from so-called the water spots. So if there was water on the slide before, maybe they rinsed it with water, and when the water evaporates, then it leaves behind those water spots. And that's basically, these are what I think might be calcium carbonate crystals. And that's not supposed to be there. It doesn't look nice, right? So what I have done is I have actually taken a whole bunch of slides and I cleaned them using vinegar and I simply rinse it with water. And I'm going to put that in. And that's how the clean slide looks like. Yeah, it's not perfect, but it's way better than the one before. Okay. Yeah, so just wanted to mention this, that slide cleaning actually, even before first time use, is actually also sometimes important. Okay, so let's go back to the desk view again. Okay. Occasionally I'm reading the comments here. Okay. Hello, hi from England. Any experience using the Nikon sensor? No experience. Industrial. The reason I'll comment on that later. The reason why I'm using industrial HDMI is simply because when I'm doing live streams, I do not need the high resolution, and for this reason, I've got an HDMI. Yeah. How are slides cleaned in hospitals? Good question. I can tell you the following. I would not be surprised in hospitals if the slides were not cleaned at all. Okay, the reason is because you need to have everything must be highly standardized, and it's infectious material. Yeah, so I basically assume that probably, especially in hospitals, it's probably not cleaned. Yeah. When I used to work in a microbiology lab, we used to autoclave everything because there were bacteria on there, so everything had to be autoclaved. This means it had to be heated up very highly, and basically steam pressure cooked. and then everything was disinfected. Huh? So yeah, let's see. You see, I'm actually not seeing a lot of green stuff here, so I'm going to try it again, because maybe, yeah, this is better. Okay, so, and then let's see what we're able to find here. I'm now getting a cover glass, and in the meantime the other slides are drying. Okay, and cover glass goes on top. Okay, and yeah, cover glass is not floating. That's okay. Let's have a look. Okay, let's do again a little bit of exploration here, and the scope view as well. Okay, and yeah, that's the 40 times. Let's refocus, and that's what we're able to see here. Okay, so I think it's a good idea to make it a little bit larger. Let's turn up the light, and we can actually go for exploring. Yeah, nematode worms, of course. Yeah, yeah, single-celled organisms here. So there's a question here also. What's better for I'll talk about this. There's a question here: inverted microscopes or not? I've received recently a email question about this as well. Somebody wanted to buy an inverted microscope. They are used for entirely different things. Yeah, and I'm going to maybe later on. After approximately an hour, I would like to maybe give it a little bit more time, because it's a fairly common question. Short answer is I think that an upright microscope like the one that I have is better suited. The reason is because inverted microscopes are actually designed to be used in cell culture. Okay, but then again, yeah, you still might get useful results, yeah. But generally, if you really want to go with very high magnifications, you probably want to use an upright microscope. Yeah? So let's have a look. Let's zoom in a little bit. I mean, I've already seen before in the afternoon when I did this a couple of dividing cells as well. Yeah, yeah. This guy almost like, yeah. So. And the question is now, are we able to make a permanent mount of this, right? So this is kind of the question here. And one of the things is that the drying process that we're going to do is actually a little bit of an issue, because when specimens dry, they might lose their shape, and of course they stop moving. Obviously, right? And we can already see that the movement is actually quite important to even see if something's there. Yeah, because if I just go through the slide here and I see some movement, then it's easy for me to focus my attention on that. And we're going to see that once when we're looking at permanently mounted slides, yeah, it's more difficult to actually distinguish the living things, yeah, because yeah, they're just staying rest and put. Yeah. Yeah, okay. Nimbot is asking, I ask about the inverted scopes because I can find some great deals on them. I will give you a short answer. Microscopy for me and probably for you is a hobby, right? And my suggestion is if you find a good deal, get it, use it, learn from it. Okay, if you discover that it works for you, then great. If you discover it doesn't work for you, then get another microscope. I've got several microscopes already. Yeah, I wouldn't say that I really collect them, but there are different microscopes serve different purposes, right? So my suggestion generally is especially because it's most cases a hobby and you do not have any specific requirements. Right, we're doing this for fun, ultimately, and for discovering. For this reason, I would simply say, if get it, experiment with it. If you really don't like it, try to sell it again, or not. Keep it, upgrade it, tinker around a little bit, get another one, yeah, and learn in the whole process. Right. So I would not overthink it too much. right? The only case where I would overthink it a little bit too much is if the microscope is too expensive. That's just, then you have to, obviously you have to think a little more about it. Yeah. So yeah, Olympus CX40 missing objectives. Honestly, the CX40 is a modern infinity microscope. You shouldn't have any issues getting objectives. Yeah, I would say probably even new ones from the company Olympus, because for the CX40 they should actually still be making the objectives, to my knowledge. Yeah. But it's an infinity microscope, so you have to actually get Olympus objectives. Yeah. Okay. Yeah. So that's there was also a comment a little bit on the prism. Yeah, I've got the so-called differential interference contrast (DIC), so I can actually play around a little bit here. If you want to see the natural colors, then it's bright field, right? So these are actually the natural colors. So you see that there is definitely it's green, right? So because of the algae. But as mentioned, I like to give it a little bit of blue, a little bit, simply to make it look nicer, to imitate the blue color of water. Okay, yeah. I'm just for your info. I'm still waiting for it to dry. It's slowly drying. I've got it now at 55 degrees, so I'm slowly drying the specimens. And you know what? As we if we can actually go up. Sometimes people are asking me what's the ideal magnification. Okay, and right now it's a 40 times objective, and you can see that because of the movement and the fairly limited field of view, then of course it's more difficult to actually chase down the microorganisms as they move. So I generally say that probably around 20 times, this one over here, 20 times is probably a very convenient or very common. I mean, what do you mean common? That's one of the magnifications I prefer using when observing water samples. Yeah, because you do see sufficient detail. At the same time, it doesn't magnify so much that it's difficult to actually chase around the organisms. Yeah? Let's have a look at those ciliates over here. 10 times, like over here, right? Still works, but it's already kind of small, right? And so 20 times generally is quite good. 40 times, as we've seen, I have to adjust 40 times as we've seen is already narrows down the field of view. So it's pretty good for stationary objects. Right. But as you see, as soon as something is moving, it kind of moves out of. So another thing is the following. Let's just take something stationary over here. I have now a 60 times. I'm not going to be using oil immersion. I don't like oil immersion so much, right? And I just want to demonstrate something to you that some of you might already know, some of you not. But my 60 times objective has a special feature to it. It has this, and maybe now just want to check what I'm doing up there, right? It's called I'm able to turn it over here, and look what happens to the image. Yeah, you might see that this is not focusing, right? But this is an adjustment ring to adjust the thickness of the cover glass. So the 0.17 over here, that's 0.17 millimeters for the thickness of the cover glass, because not all cover glasses have exactly the same thickness due to manufacturing tolerances. And this allows you to compensate for that to get the maximum and best resolution. Yeah. So this is some kind of an adjustment collar that this specific objective has. Honestly, I've been never really using it. So, but this is something people who need the highest resolution and who don't have this, what they will sometimes do is they have a caliper to actually measure out the thickness of the cover glass and choose those cover glasses that actually have the 0.17 millimeters. Okay. So this is let's go a little bit lower with the magnification, and I want to have a look at another sample as well. Okay, it's dry now. Very good. Okay, at least two of the slides are now dry. Let's have a quick look at those. Okay, I just want to show you this here. So that's the one from before. I'm going to now put this one here also under the microscope without cover glass, without anything. Okay, and let's have a look here. I have to refocus, of course, because there is no mounting medium, and this is how it looks like. Okay. Yeah, so this is basically in air now. We can actually see the diatoms quite nicely. And those permanent slides are then good if you really want to go up with the magnification and want to look at some details. But now, as promised, I mean, I don't know. I'm not able to find all of those ciliates and whatnot. Yeah, I'm not able to find them anymore because they're not moving. And it could be that maybe they've also lost their shape during the drying process. Yeah. So you see, that's a little bit, yeah, a disadvantage, so to say, yeah. So that's again the. Yeah. Let's have the following. Let's try this. This is now with the without a cover glass. Let's see if by adjusting now the ring here, if this actually does make a difference. Yeah, you see that this does look a little bit more blurry. No, okay. Yeah, you have to adjust this until you get the best resolution here. Yeah. Okay. Yeah. Yeah, the critters ran away when it got too hot. Yep. The, unfortunately, they did not have a possibility to swim away because, obviously, yeah. But that's indeed one of the problems because those diatoms that you see here and the algae, they have a cell wall, right? But many of those single-celled microorganisms, yeah, they're quite soft and they shrink when they dry. And they lose their shape, and then you cannot find them anymore, right? So I just wanted to, yeah, the sweet slides are now, how do you say, dry? Let me quickly turn this off. Yeah, I'm going to simply put them a little bit to the side now, so that, yeah, it's over here. So let's do the following. Let's go back here again. So that's again the fresh sample, and I would like to do now the following. I would like to now take a sample from the algae and put that under the microscope. Okay, let me see where the desk view is here. So I'm going to put the other one back again. And where is I now need some tweezers. Ah, yeah. While I'm doing that, people, I wanna inform you that indeed I'm not only doing live streams, I'm actually also making videos and YouTube Shorts. And I just recently, yesterday or so, I uploaded a video that I made where I put the wood of a 1,000-year-old oak tree under the microscope. It's right in this channel. It's uploaded it yesterday. And yeah, I show you how I prepared some oak wood, and yeah, I talked a little bit about the tree and so on. And the video is getting very, very few views. I don't know why. It's interesting. I'm getting more views on the live stream here than on my regular videos. I don't know why this is the case. I just want to advertise it a little bit so that maybe if you're interested, maybe you can also watch maybe some of my other videos simply to get the views up a little bit. Yeah, so it is in the same channel. If you just click on the videos, you're able to easily find it. You see, that's one of the things here. You have to separate all of those algae here. The advantage of those algae is that sometimes, if you take those filaments, many microorganisms sometimes are attached to those filaments. The disadvantage is that sometimes the specimen is a little bit too thick. So let's take a little more water. Let's drop on a cover glass. Okay, and let's see what we're able to find here. And now then you will probably see the advantage of actually filtering it, because usually this is very dense and difficult to see. Just a second, the scope view. I'll have to wait until the camera starts to, yeah. Let's go down with the magnification here. You see, and that is the reason why I filtered it, because this is way too dense, right? Yeah, you do see a lot of movement here yeah, there as well. Those algae, they produce oxygen, obviously, and that is the reason why microorganisms that are moving around, they like to be next to them. It's not only a source of food, but they produce quite a bit of oxygen as well. And movement requires a lot of oxygen, because the oxygen is needed for cell respiration as an energy source, and for breaking down organic substances, it needs oxygen, and those microbes, they generally, especially those larger ciliates and so on, they need a lot of oxygen, right? But as you can see, more difficult to actually see the individual cells because the algae cover them up, obviously. Yeah. So, and that's the reason why I was kind of stirring them first and filtering them, because this way they become more easily visible. So a whole bunch of diatoms again. What I have done and what I will do still is I will take a water sample, and I will take a water sample and add a crushed wheat grain to it. And this will work as a food source, and then ciliates, specifically Paramecia, are going to start multiplying quite a bit. Yeah. So because bacteria will start to feed on the cereal, on the wheat grain, and then the larger microorganisms, the ciliates and flagellates and so on, they will feed on those bacteria. You're essentially establishing a small food chain, and then you have a highly concentrated sample of Paramecia. So what I'm going to do is I'm quickly going to read some of your comments again. Okay. Let me quickly see. There's a question here. If a ciliate dies, does it burst open because it does not maintain the internal conditions, osmotic pressure? That could be indeed one case, and the reason is the following. Ciliates like Paramecia, for example, they have a so-called a contractile vacuole, which constantly pumps out water, right? And now if the cell dies, and the contractile vacuole stops pumping, then water will move in by osmosis and the cell might burst. So this is probably one of the reasons. However, there can also be other reasons why they die. So let me quickly have a look here. Again, some of the comments. Yeah, there is a question by Light about soil microscopy. I've honestly received a fairly long email, also by someone, which I unfortunately did not answer because I wanted to make a separate video of this. There was a question of what happens if you put soil or soil water under the microscope and you don't see any microorganisms. So what's going on? And the reason is why you don't see any. They are there, definitely. They're primarily fungi and also bacteria. Those organisms that you see over there, they are the flagellates and ciliates and so on. They actually need water to move around, right? And you won't find them so much in more or less dry soil. Yeah? So even though they're there in soil, they're different kinds. Yeah, this is an ethical question. Arventis, do you ever feel ethical problems with disposing of the specimens once you're done with them? I always feel bad, especially if they are more complex animals like arthropods. This is an interesting question. Actually, I wanted to answer those off-topic questions later on, but I'll answer them now as well. I will answer this, but it's an important question. It's ethical questions about basically killing microorganisms. And I will tell you now the following. I don't even know where to start. Those microorganisms, they do not have a nervous system. Okay, so number one. So they won't be able to feel any pain. They don't have self-awareness. They might appear to be intelligent. I deliberately say appear, like they're able to respond to stimuli and so on. They move towards light, they move towards food, oxygen and so on. However, they do not have a self-awareness. Okay, so these are automatic processes that happen without any consciousness. So that's number one. So I have no ethical problems in in actually killing those. Number one, I understand it might be something different with insects. I will tell you the following. It's summertime right now. There are plenty of insects and spiders and bugs and beetles in my apartment that I do not want to have them in here. I always catch them and throw them out the window. I never kill them. Yeah, just because I don't like doing that, right? But still, yeah. So, and now to the point. I remember there was once this was several years ago, and this just illustrates a little bit how biologists think. So this is going to go a little bit deep now, okay? There was once in a zoo, they had to kill an animal. I think it was a giraffe, right? There was a reason why they had to do that. And the people were understandably very much against that. So, then they asked the director of the interview, they gave an interview of the zoo director. The zoo director then said is what the people don't, so I'm paraphrasing right now, what the people don't understand is I'm not worried about the individual animal. What we have to worry about is the gene pool or the population. That's how biologists thinks. Who cares about the individual animal, right? Important is the species survives. Right, and that's a little bit also over here. What is important is the biodiversity that we keep those, and not the survival of the individual cell. And that's a different way of thinking about nature. It's the survival. What is important is the survival of the species, and but that's a little bit, how do you say, difficult to communicate to people who are understandably feeling sorry for an individual animal that they have a personal relationship to, right? So, and what I'm saying is that microbial biodiversity also, of course, is important. However, what we have to be aware of is it's not the survival of the individual cell, but rather the survival of the whole species or population or the gene pool, right? And when we are worried, understandably worried about biodiversity as such, you have to understand that every time when a pond dries out, right, we are a lot of how do you say organisms die, and for me the death of microorganisms is a little bit. I know it's an analogy right now, but if I scratch myself, I'm killing off thousands of skin cells, right? And I don't feel sorry about those either, right? Because they're just basically a part of a larger whole. And you have to see the individual cells here not as individuals, but also as members of a larger. What is this here? As members of a larger community, right? That's what is this here? Is this some kind of a worm or something? Yeah. So I was drifting off a little bit here. Yeah. But I get those ethical questions here, right? Because indeed this is also something that appears every time when, for example, I'm a teacher, and every year we have a so-called an insect project with students where we are. Hiring an entomologist. He's an insect specialist, and together we're catching insects and we're identifying them. And the entomologist actually, before we actually kill the insects and put them under the microscope, he's sorting through them to actually remove those insects that are actually protected. Yeah. So yes, we are also, as biologists, have these ethical standards, and it is important that we have those. And if you watch my video about the tree, which I basically made, I was looking around and said, which sample of the tree can I put under the microscope? Of course, I could have easily cut off a small, tiny piece of the bark of the tree. I could have it wouldn't have harmed the tree, but out of principle, I did not do that. Yeah. So I was I'm wondering, what is this here? What I'm looking here? This looks like a worm or something. You can see the peristaltic movement. Yeah. So that's let me have a. Yeah. So what I'm going to do now is I'm quickly going through here. Have you found any hydras or water bears? Yes, both. Yeah, hydras are found in basically in when you collect. If you want to find hydra, go to a pond and collect some water plants. Hydra like to sit on a surface, right? So you have to collect water plants, and they're generally in the biofilm. There are plenty of things in there. Water bears, yes. In moss, you have to find a proper moss sample, however. Yeah. So let me quickly go through the comments here. Okay, you know what I'm gonna do? Yeah, some people joined in. Yeah. So what I'm going to do now is we're now going to, yeah, after my little philosophical excursion, let's go back a little bit to the slides that I just prepared. And now let's try to make a permanent mount. So here they are, 1, 2, and 3. Okay, they are now completely dry. And what I now need is the following. I now need a, again, my little, how do you say, coffee warmer. And I'm now going to show you the following. However, I'm not going to be using this. I do want, because I know that it works, but I do want to use the slightly more advanced one here, the glycerin jelly. So again, for those of you who are new to this is basically a PVA glue. It's called Elmer's PVA. It's a clear glue where I added a little bit of water, and it is a water-based mounting medium. So it's actually very cheap. And I would, if you want to make permanently mounted slides, start with this one here. It might be a little bit more difficult to get the clear glue because nowadays mostly they're white glue. But if you look around a little bit, you're able to find it. You add a little bit of water, and this should do the trick as well. Yeah, works, right? However, this is not because it dries and becomes completely dry, it might exert some, how do you call this, some stress or tension on the specimen. Yeah, so it completely dries out. This one over here, glycerin gelatin, remains basically semi-solid. So it retains water. It will not completely dry. And therefore it's more, how do you say this, not as harsh, not as, how do you call this, yeah, aggressive to the water samples. Okay, yeah. So they're going to reabsorb some of the water again, and yeah. So the only problem with that is, or not the problem, the difficulty rather is how are we going to do that? Because if you open it up, you're going to see that actually it's solid, right? And what is important to understand is that this one has to be, you have to melt it, and when it's molten, you put a cover glass on top. And then you let it cool, and that's it. And then you have to add a ring around it, which I'm also going to show you. Okay, so the thing is the following. I have to warm it again, and we have to be careful because this glycerin gelatin should not become warmer than about 40 degrees. Okay, otherwise it's going to, yeah, it might not re-solidify. Yeah? So we're going to pre-warm the slide. Your 55 degrees should also do the trick, right? And then what I do is I'm taking a small sample of this here. Yeah. And I take this, I put it directly on here, and I wait until it melts. If I can only get it off. This is a lot. This is actually quite a bit. Now the thing is, if you smell it, it smells. It has this typical hospital smell. I don't know if you know what I mean. And this hospital smell, that's actually a substance called phenol. So they added phenol to this, and this is a disinfectant, right? Because you don't want this to turn bad. You see, it's already starting to melt. Okay. So it's only 40 degrees and it's already starting to melt. The difficulty with glycerin gelatin is that it likes to make a lot of bubbles. Okay, that's a little bit the problem. And if you're kind of wondering why it's pink, and that is because they've added a stain to it called fuchsine. Because this glycerol gelatin is actually there for staining pollen grains. Plant pollen, and I'm just also using it for water samples. Okay, so you can see that it basically has now melted. What's the correct way of saying it? And I like to use now round cover glasses. I'm just unpacking those. And I just see that unfortunately, yeah, they are also a little bit dirty. So I have to dry wipe them. And it's going to become clear in a couple of minutes of why I'm actually using round ones. So I'm dry wiping them. So, and I might simply drop it on here, or where do I have this? Maybe you remember this from last. Maybe you remember this from last week when it was kind of extracting. So I'm just trying it like this, and then I'm trying to place it more or less on here. And I let capillary action do the rest. You see bubbles are there, okay? That's always a general problem. Yeah, it's not a problem. I mean, then we'll look at those places which basically don't contain bubbles. Maybe we can press down a little bit. Maybe we're able to squeeze out some of those. Yeah, I've also used quite a large amount. Okay. Yeah, this is actually way too much because you see that it's kind of spilling out on the side. Yeah, so next one. I'll put it over here on the side for cooling. And now I'm definitely going to use much less. Where is it? Here it is. So let's take again a small sample here. This was way too much before. So we wait again for it to melt. I mean, if it's 40 degrees, so it's just barely above body temperature. Okay. In the meantime, it now melted would be correct. And once it has melted, it has become molten. Thank you. Here we go. Yeah. So, hello from Utah. I'm just as it's melting. Gastrotricks seem very conscious. Well, the whole thing question about consciousness that is getting complicated, right? Yeah, you need to the best of our knowledge, a nervous system for consciousness. Now, I know, not a debate with artificial intelligence and so on. Yeah? So let's give it a try now again. This is much better. Okay. Yeah, it's actually too little even. Let's warm it up again. Yeah, you see, getting the correct amount is critical here. Okay, so, and this one over here. So, and we're going to try. There is also another possibility. In the past, what I've done is I actually put it on the warm coffee warmer. But I found out that this takes quite a fairly long time. And indeed, this should also be possible. Or if this doesn't work, we're just going to go in here and just take a small part out here. Here we go. The glycerine that is in here actually prevents it from completely drying out. Okay, and yeah, as it's melting, as it's melting, I've got over here, what you see over here, these are, if you watched my video about the oak tree, yeah, these are the cross sections of the wood of the oak tree, which were embedded, which I soaked in glycerine, in pure glycerine, and it didn't dry out, right? So I can actually keep them in here. Yeah, and the glycerin also prevents the growth of other microorganisms because it will remove water from them. So here we go. Okay, so, and then we're going to have a look at those. And then I'm going to quickly also show you some slide ring, slide ring oak chips. Yes. Yeah, this one takes a little longer. Maybe it's not really making good contact with the slide. Could that be? Okay, yes. Usually it's like this. It's got to make good contact, and then when it makes contact and starts to melt, then it should be faster. Could also be that in some places the coffee warmer is warmer than in other places. Okay, now it's starting to melt. Over the next couple of days, the red pigment might actually start to stain some of the organisms. So that's also something that I'd like to observe. So I'm going to dry wipe this again. And let's try it here. Yes, I think now I got the amount correct, more or less. Good. So we're going to get this removed because I do need space. It's really, my workspace over here is really extremely, how do you say, constrained and crowded. So here they are, 1, 2, and 3. The one, which is a little too much. Let's have a look in any case. Okay, let's remove the old slides here. Some people ask me, where do I get the materials from? The Brunel microscopes in the UK is a very good source. Not sponsored, nothing like that, but I ordered from them already several times something. And so that is now let me see if this, here we go. See, these are the bubbles. Okay. And that's actually, this actually shows a couple of interesting things here. A couple of things that we have to get sorted out. Not so nice. I just want to show this to you because this is something that we've seen already several times. Look, let me turn on the arrow. Not so nice. I mean, those round bubbles is clear, but you actually see in here, inside the cells, these are also air bubbles inside the cells. So the mounting medium, the glycerin gelatin, was not able to fill out the cells here. Yeah, so this means that there is still some air caught in here. Also, for example, in here as well. Air bubbles. So that is one of the, here a lot. So that is one of the things that, is unfortunately something that, we know. Let me turn on the DIC again. Here we go. Okay. Yeah, so that is a typical, how do you say, sign that there was air inside the algae. So one of the things that I might want to try, maybe not today, but maybe not to dry it completely. That's important. Right now, on the coffee warmer, I dried it completely. Maybe if I leave it a little bit moist, then maybe those bubbles won't be there as well. Okay. Yes, a centrifuged another probe. Is that to remove plant matter or for getting a higher concentrations? It was actually for getting a higher concentration. So what I've done here, where did I put the tube? Here it is. right here. This tube I filled up with water, the one that I filtered, and then I was spinning it for a couple of minutes, and then on the bottom I had a pellet here, but I found it's not necessary to do that because it kind of naturally sedimented. I'm going back over here to the desk view. It naturally sedimented out in here. Okay, now I can see it. Yeah. So this was very green, the water, but yeah, it kind of sedimented out, so it was not necessary actually to centrifuge it. I'm also going to show you something else what I have, but yeah, I'm just going to show you later. I've got a plankton net here as well that I've not used yet. Yeah? So let's go back here. But the centrifuged one looks exactly like the one over here. So I'm going to now remove the, how do you say this here? And there is something that we now should be able to see. Look that some of them start to turn pink, some of the, whatever this is. Yeah. So you see that some specimens here, some cells started to absorb now the red pigment. And this is something that should actually increase with time. Okay. So another thing I want to show you. Two things that I want to mention. Look, when I focus, do you actually see that there's movement going on here? The reason is because this one is already pressing against the cover glass. And what you see over here, this is a so-called a conidium. That's the spore of a fungus. Very, very typical. Yeah, so that's a fungus. Yeah, it's called a conidium. Yeah, yeah, and the reason why it's moving is because it's pressing it's pressing down on the cover glass. Yeah, so you see that some of these structures start to absorb the red pigment now. So, but this one I think is the thick one. Yeah, so now let's have a look at the very thin one over here. That's the one where I used very little. Okay. Yeah, same issue with the bubbles here. Yeah, a big bubble over there. So what do I learn from this? The next time I'm not going to completely dry the specimen. I wonder what those dots are in the background. Yeah. I wonder what those dots are. Difference between conidia and sporangia. Or a conidium a specific type of sporangia? Let me see this over here. You know what I'm going to do? Salt crystals, I doubt that these are salt crystals because this is water-based, so it should actually dissolve. But over here you can actually see that, yeah, some of those structures, I don't know what it is, starts to absorb the red pigment as well. So, you know what I'm going to do? I'm going to play this back to the community. Difference between conidia and sporangia. Maybe somebody can find this out for me, for us. Because conidia are sometimes also referred to as the spores of a fungus. Thank you very much for the question. And the spores are actually made by spore-forming structures which are sporangia. Yeah. Yes, sporangia are spore-forming organs. Yeah. Okay. So I wonder indeed what these round things here are. I mean, salt crystals. I mean, this is a freshwater sample. Yeah. So let's have a look at the other one. And then I'm going to do the following, and then I'm going to quickly try to ring a few of these slides, or at least one or two of them. Again, look at this. That's interesting. Again, those things here, what are they? Something almost seemed to have precipitated. Yeah, again, one of those conidia over there. Yeah, yeah, you get the idea that. Yeah, what I'm going to do now is the following. We were just going to see how this responds or reacts over time. You can see that as the time passes by, it will absorb more and more of this pigment. I've done this already also with spores, and those spores will really turn very bright red over time. Yeah? The glycerin crystallizing, I mean, I cannot imagine the glycerin to crystallize because glycerin actually is a liquid. And glycerin actually absorbs water, calcium deposits possibly, yeah, but I did not see those before. Yeah. So evidently, when I dried the sample, something deposited that we're seeing right now. Yeah. Yeah. So that's another thing, evidently that yeah, and somehow it did not redissolve again when we were adding the mounting medium. Let me see. So what time is it? Okay, it's 1 hour now. What I'm going to do now is I'm quickly going to do a demo. Let's see how well I'm actually able to manage this. I'm going to ring now a few of those slides here. I'm going to show you what this is actually like. I would like to. So here are the slides here. Just want to show you something. Some commercial slides. And just a second. I'm gonna get my slide box. So here we are. Let me show you something here. So these are commercially prepared slide boxes. You're going to see that yeah, some of those slides have this ring around it. And this ring here, and here are two more. Okay. Yeah, so these are two algae. Okay, there's Oscillatoria and Spiragara. So these are two algae. So I suppose they were also mounted in glycerin gelatin. And the reason why you have to add a ring like this is to make sure that the cover glass is properly attached. So that's one. Okay, that's one thing. And the other thing is it also prevents, when you have changing environmental conditions, from actually water and moisture moving in and out of the slide. Yeah. So you add a ring around it. And what I like to do is I like to use nail polish for doing that. And I would simply like to show it to you. I'm not saying that this is not going to be very perfect, what I'm doing. And as a matter of fact, over here, this slide over here, which does not contain, which is basically has some air beneath the cover glass over here, because it did not use enough of the mounting medium. Some of the nail polish might actually also go beneath the cover glass. Okay, so I'm just saying, yeah. So and over here, yeah, and over here, I wonder if we're able to I don't know if this is actually. You see how the cover glass actually is a little bit unstable. Yeah. So this one is probably not the best. This one over here is probably not the best slide because I used too much mounting medium over here. Okay. You don't want the mounting medium, the glycerin gelatin, to actually touch the the nail polish. You might want to remove this. Yeah. Or I would say, yeah, I would probably say maybe just redo the slide. Yeah. So this one is probably not a good one, right? And this one over here is a little bit better. And this one over here contains too little mounting medium. It doesn't matter. It's just a demonstration. And what I have here now is the following. Let me remove the water. Let's move this up here a little bit more. And I'm just going to start with this one over here. And this one is a so-called a slide ringing table. So you can spin this. I think I made some time ago. I've been using it, yeah, always. So this means that I made some videos on how to use this as well. So now the tricky thing is I really have to make sure that this one is all the way really properly centered. And this is a little bit difficult because I have a difficult time actually seeing the cover glass. So a little bit difficult to see that. Yeah. Here, no, now it's, yes. So, and what I'm going to do now is I'm not going to use a brush. I found out that a brush is not going to give me quite as nice of a result, but I'm also going to use a pipette now. And where is my nail polish? So this is just a standard, yeah, nail polish and no brush. Okay. I'm not going to use a brush. I'm going to now simply take my pipette and now this requires a lot of patience, okay? And up. And you kind of hope that this is actually working or not. And if it's not working, then I have to add a little bit more, how do you say, solvent to it, right? So that's basically number one. Yep. And you allow it to dry. Maybe I should have added a little bit more. Yeah. And you allow it to dry before putting it under the microscope. And then you can try it with the next one. And this I'm also using to actually make some spacer rings. So sometimes I've done this already over here. Look, it actually looks nicer. So there's no cover glass on here. If you do that, then you actually have a small little spacer and you can basically have a larger distance between the cover glass and the microscope slides, for example, for observing larger organisms that you don't want to have crushed. So I need to use a new tip. And you know what I'm going to try now? I'm just going to try to make this a little bit larger here by cutting it away. So maybe this is going to work better. Maybe it's going to flow better. I don't know. Bad. Yeah, kind of. It requires practice. Okay, I hope you kind of get the idea. Usually, yeah, it will look a little bit better after a couple of minutes after the nail polish starts to spread. Yeah, and then when it dries, it shrinks again a little bit. Yeah, but as long as you have some kind of a proper seal, yeah, it should be fine. Huh? So. Yes. Pre-marking the round cover glass with a black felt tip pen. We already talked about this before. I forgot. So, look. The idea is the following. Yes, thank you for reminding me. What you do is the following. You take a cover glass and you take a pen. Yeah, and you just lightly, yeah. You might not be, yeah, you can actually see it here. Yeah, you can actually see the edges much better. Yeah. Here we go. Yeah, I should have done that. Thank you for reminding me. Yeah. Yeah. So then you're actually able to see it much better. Yeah. What I've done here is I've put a black ring over here. But then, of course, I'd use some alcohol to remove it again, and then you should be able to see it much better. Yeah, by placing it on here. So let me quickly have a look here. Yep, this was about the crystals here. Is this happening on all three slides? I don't. Are you referring to the crystals here? Let's have a look at the other one over here. Let's have a look at because this one I was not quite sure whether we were able to see those crystals. Scope view. The arrow we take away. So let's have a look. This is the one where I have a lot of, where I used a lot of, yep, here as well. You see, it's quite nice, interesting. What we see over here is we see that over here, this was where the drop of water was, right? And over here, where it was not. So there where the drop of water evaporated, this is where we have those crystals here. A glass paint and a small artist brush. I tried using an artist brush as well. I did have the following problem a little bit, and that that the nail polish started to dry very quickly. So I have to probably make it more fluid by adding more solvent, like acetone. Then it might actually also work. But I've by trying it with a pipette, I've also in the past gotten decent results like this. Yeah. So yeah, but this kind of shows indeed here that there are basically some crystals that have formed. Yeah. And a lot of bubbles. Okay. Yeah. So let me have a quick look again. If there are any, yeah. How do I prepare? You know what? I'll do now the following. I received again your questions. I will quickly go through your questions if I have not answered them yet. And then I'll answer a couple of other questions here because the preparation of small insects and so on is also something. So let me quickly, let's put this away here carefully. Okay. Yeah, so no, I don't have experience with that, unfortunately. Yeah. How are slides cleaned in hospitals? I would say probably not. The reason is that also in hospitals you're working, I think, for two reasons. Because you want to have really highly standardized conditions. Number one, and number two, if there was actually infectious material on the slides, I'm not quite sure whether they actually you want to actually clean them. So I can imagine that they're probably thrown away. Yeah, hydras I have found in pond water. A colleague of mine who I work with, he's got an aquarium, and he said is that those hydras are all over the place. That it is actually sometimes even become a problem, right? And they're growing on the inside of the aquarium, on the glass surface, and so on. I found hydras already before on plants from a pond, and I've been feeding them. So I actually also added tiny water fleas, and you can actually see that how they're actually catching them and how they're actually sucking them in. Excuse me. Generally speaking, what to prefer between industrial HDMI camera and second-hand cheap mirrorless cameras for microscopy, video monitoring, and video grabbing. Okay. Now, generally, if you use an industrial HDMI camera, like for example, not the one that I'm using right now, but I've got one for my stereo microscope. They have the advantage that they are C-mounted. You can connect them, in many cases, directly to a microscope. They have a fairly small sensor. And basically you then get an HDMI signal, which you then have to convert to USB. Or you can directly connect an HDMI monitor. So you can actually do with an HDMI industrial camera, you can do basically you can see the picture on a monitor and you do not need a computer. That's really nice. Right? So it basically you switch on the monitor and you can actually see the picture on screen. Now mirrorless cameras, what they have is they have a larger sensor, However, there is a difficulty here, is you need a proper adapter system, probably also some intermediate optics to actually connect the mirrorless camera. So generally, they're, because of the larger sensor size and everything, they're gonna give you better image quality. However, sometimes you don't need that. Unless you're taking pictures. When you're live streaming like I'm doing, is I don't need a higher resolution because yeah, it's not needed, right? So it really depends a lot on what you actually want to do. Yeah. And video grabbing, again, here the question is what quality do you want? And if you now have a mirrorless camera and you have a USB 2 connection, for example, then you're not going to get the frame rate. Right, so then you might get the resolution, but it's not going to be a very smooth image because the USB is limiting. So you see, it depends on so many factors that I can actually not say which one's better. It really depends on what you want to do. Yeah. Any thoughts on what's best for pond microbes, compound scopes, or inverted? Yeah, that's the thing that I wanted to talk about. Inverted microscopes, and I don't have yeah, here I've got it. Inverted microscopes have the basically the lens on the bottom. Okay, I don't have a spare objective. They have the objective on the bottom. A light is coming from the top so that you can put a petri dish directly, on the stage. The reason is because in cell culture, what you have is you've got, for example, human cells directly growing on the surface of the petri dish. Now, having said that, basically, if you've got pond microorganisms, what you have is you have got them swimming around. You need a cover glass, and the thing is that especially at high magnifications, look, it's much thicker than a cover glass. So this means you actually need optics that are, if you want to get the resolution, that are designed for basically for thicker, larger distance. Yeah. So if you really want to get, I think if you really want to get a high resolution or a high magnification, you have to go really close to the specimen. And in this case, you need to have a thin cover glass and not a thick one, thick petri dish. Yeah. So the purpose of inverted microscopes is different. Having said that, however, if you've got some kind of a pond water sample and put it directly on here, and yeah, it might also be easier to observe, however. Right. So you see, generally it has pros and cons, but it's not just that the optics is on the bottom side, but actually the optics are also different. They have large working distance, as it's called, because the distance is further away, right? Because they're designed for petri dishes, right? So in that sense, I cannot really say that they're automatically better or worse. They have, again, different areas of applications. Yeah. Yeah, I just got back from a hike, past lovely stream full of algae, but didn't have any collecting equipment. That is indeed one of the things where I then recommend that you have, look, I can do it like this here. These are centrifugation tubes. They're actually disposable. They come in different sizes and shapes, right? Yeah, and I always carry a few of them around, yeah. Simply if I find a sample, water sample or not, doesn't matter. You just basically can put them in here. It's watertight as well. Yeah. I also use this here. You can actually put some tools in here as well. Yeah. Something like this. Yeah. It doesn't work because it's now too wide down here. Yeah. You get the idea. Yeah. So this is, and they're quite lightweight as well. How do I get that beautiful blue background? Yeah, that's DIC. That's, I have a little bit, I admit I have sometimes a little bit mixed feelings about that because people really, look, yeah, it looks really nice. Yeah, you can change the colors around and it's polarized light, right? But I know that DIC is expensive, and in most cases, what you do is you just basically want to use bright field. If you do want to get colors, oh my gosh, all of those bubbles. If you do want to get colors, then I do recommend that to probably use color filters, right? DIC looks nice, but again, it destroys the natural colors, obviously. right? It's a bright field like you see over here. That's actually how it really looks like. So the ethical problems, I talked about those. You see, yeah, you see, if you observe, I don't know, I'll be honest with you, I've kept sometimes those Iolosoma worms in a dish for, I don't know, days, weeks even, right? Of course, you kind of feel a little bit, I don't know, and you keep on watching them over and over again, dumping them down. I don't know, it doesn't feel right. So what I do is I usually do put them back into a glass jar. Huh? But honestly, I also have to say that. I mean, it's good to have ethical concerns, but I think it's also important to have really ethical concerns. And I actually tell my students that about ethical concerns that actually do matter. And several years ago, and you can still find that video, I made a video of a paramecium that accidentally got crushed by my cover glass. And I made a video about that. And this video went a little bit viral. And I had a lot of people commenting and being pretty upset about this, right? About the paramecium, the single-celled organism being crushed because of the evaporating water. And yeah, some people actually says they're going to file a complaint and to the animal rights and whatnot. Look, we've got a lot of environmental problems in this world, right? Where we really should be ethically concerned. And what I also tell my students, I'm a biology teacher, right? I says, if we want to be ethically concerned, I think that's a good thing, but let's be ethically concerned about the things that actually matter. Okay? And those microbes in my jar, I do not feel sorry about them. They don't have a nervous system. They will outlive us all as a species, as a population. But deforestation, the problems with climate change, all of these other issues, pollution, plastic in the ocean, they are real ethical issues that we have. Let's save our energy for those problems. Having said that, I get the point. I get the point because I too don't just kill microbes that I basically have observed under the microscope, right? Yeah, so I get it because but sometimes I have to say, let's worry about the real things, you know. If a cilia dies, does it burst open? That's the contractile vacuole, right? I tried looking for microbes to look in the soil. Soil. What you have to do is the following. You have to take some soil, you have to add water, and you have to let it stand, and this will basically dissolve some of the microorganisms out. However, having said it, it's mostly fungi and bacteria, and they don't always move. And they're difficult to see because of all of the soil particles, right? So that's a little bit, microorganisms are there, but difficult to see. Yeah. The chloroplasts of the cytomes have also shrunk. Yes, quite well possible because as water evaporates, the cell contents also shrinks, and the diatoms have a hard outer cell wall made of silica, so that maintains the shape, but the cell contents will shrink. If you keep wet mounts in a humidity chamber, you can watch the microbe community evolve over a week or so. A little algae in the light also makes CO2 helps. Yes, a humidity chamber is basically a plastic container, which I don't have now, where you essentially add some water and where you keep it closed so that the slides do not completely dry out. And you can basically keep them for a very long time. Okay, so... you said you centrifuged another probe. Yeah, that's the one that I've showed you, but it looks the same. Yeah? And the reason why I have centrifuged this is the following, because the algae and the microorganisms are a little bit more dense than the remaining water, and therefore I was able to concentrate those. Yeah? Is there something similar for gram tests for protozoa, or do they all absorb the same stains about the same? There is, in order to determine or to identify protozoa, specifically ciliates, there is a so-called the silver test. Wilhelm Feusner, former protozoologist, he has been instrumental in this, and he made the cilia of protozoans visible. This way of ciliates, and this way he was able to identify them. So gram testing actually is something that is used for bacteria to distinguish gram-positive and gram-negative bacteria. However, you use also crystal violet for gram staining, and also safranin, and these are very unspecific stains which will also stain protozoans. But specifically the gram test to distinguish between gram-positive and negative, that is something that's done specifically for bacteria, but the same stains will also stain protozoans. Difference, okay, we talked about this, yeah, so that sporangia, they basically form the spores. So having said that, let me quickly go through the how do you call this, the comments section. Yeah, how do I prepare small insects for microscopy? Can I use super glue to fix them on the slides? No, not super glue. What you do is the following. You use I also made a video just recently, last week or so, where I put a very old microscope slide of an insect. And what you do is the following. You take an insect and you put it into alcohol. The alcohol will kill off bacteria and will remove the water. And then you use a mounting medium, which you don't have, called Euporol. And that is the standard mounting medium to use for insects. Now, it's difficult to get. And for this reason, especially when you're starting out, use the water-based mounting medium Elmer's PVA. Superglue will not work for the following reason. Superglue is called cyanoacrylate and there is no water or no solvent to evaporate. Superglue will become hard and sticky because it reacts with water. It's kind of interesting thing. Superglue was invented during the Second World War to glue together wounds. So moisture, it will react with water, and so basically what happens is if you use superglue and then and you put superglue, then the outside will become hard because it reacts with the humidity of the air, and the inside will remain liquid. Yeah, so superglue will not harden. Yeah, so this is not a suitable mounting medium. You can use experiment with clear glues, and Elmer's PVA is, I would say, something that I've got very good results with. Yeah, other people as well. Yeah, the glycerine, we talked about that. Let me quickly check if there's anything else for me. Nope. Let's go down here. Small insects, yeah, please do put them into alcohol, or you completely dry them. That's also another possibility, right? So understanding is basically cyst, okay. Yep, no comments for me. Put a tube mount on that and give it a real good spin. Yeah, Oliver, if you pre-marked the round we talked about, yes. Try using glass paint. Okay, yep. Okay, yeah. So no more comments for me. Yeah, it's been already 1.5 hours again. Yeah, I would like to invite you, of course, to also watch the other videos that I've made. I'm always happy for feedback. I've been, over the last couple of weeks and months, I've been thinking a little bit I don't know, should I change the format around a little bit, because the views have been going down in this channel. It's interesting, and I think it also has to do a little bit with the summertime, because more people are on the outside, which is actually good, right? Yeah, but if you have any specific suggestions or recommendations, yeah, would be happy for ideas. Otherwise, what I'm going to do is the following. Yeah, I think I'm going to quickly just say bye-bye. I'm just reading if there's anything, if I forgot any comments, but I hope I didn't. Okay. Yeah, I'm going to call it quits for today again. Yeah, hope that it was interesting for you. We're going to see how those slides have been doing until next week. Okay. Not very nice. I'll just see. But yeah, you get the idea. Okay. And yeah, I wish you all the best. Yeah, happy microbe hunting as always. And yeah, because I'm on holidays now, I'm going to keep on posting videos a little bit more frequently. So the next video is already in the process of being made. Yeah, and would be happy if you are going to join me again next week, and also if you watch some of the other videos that I make. Bye bye, happy microbe hunting, and see you around. All the best.